closer than ever before if you wish.
So let's say that you want to create a custom-made, genetically engineered designer baby from scratch. You'd want your baby to be as smart as Albert Einstein, as attractive as George Clooney, and as strong and muscular as The Rock. But you would also want your baby to resist diseases and live for as long as possible. Intelligence, beauty, strength, and life expectancy are all controlled by our genes, which means you could add all these characteristics into one individual before birth. The first thing you need is a skin cell, which can be converted into a human fetus. There are around 35 billion skin cells in your body. It is astonishing to know that each individual cell has the potential to become a fully grown human being. First, you take skin cells. They could be from a male or a female. One person is enough. Then you convert them into embryonic stem cells. These embryonic stem cells have the capacity to become any other type of cell in the body. Heart cell, lung cell, liver cell, neurons, and the list goes on. And since we're interested in creating a human being, we would convert these stem cells into sperm and eggs. This can be done through a process called cellular reprogramming, and it works by feeding stem cells the right ingredients of hormones and growth factors, and sometimes a bit of genetic modification. Depending on the combination of hormones and growth factors that you choose, you can force stem cells to become any type of cell you want. Now that we have converted skin cells indirectly into sperm and eggs, we can fertilize the egg using the sperm through in vitro fertilization, a standard procedure that is currently used by millions of infertile couples around the world, and it has existed since the 1970s. In vitro fertilization allows us to create embryos from sperm and eggs under lab conditions. Unfortunately, these embryos are susceptible to genetic diseases. They could mutate and eventually experience serious health problems after birth. They may also give rise to ordinary human beings. To solve this problem, we will have to genetically engineer these embryos using CRISPR-Cas9, a gene editing tool that is fast, reliable, and precise at editing the genes of almost any cell.